Hi everyone. Now let's learn about soil. What is microorganism exist in soil? There are a lot of microorganisms exist in the soil, such as bacteria, fungi, algae, protists, and viruses. Let's take a look at the bar chart. Aerobic bacteria possess 70% of all microorganisms exist in soil. This is followed by anaerobic bacteria, actinomycetes, fungi, and other organisms. Other organisms here include algae, protists, and viruses. For bacteria, the type is autotroph, heterotroph, aerobes, anaerobes, mesophils, thermophils. Both mesophils and thermophils need a suitable temperature to live, while bacteria function as nitrogen-fixing, nitrifying, denitrifying, and digest special substances. Next is fungi. Mycelia and spores are abundant on the soil surface, while yeast mostly in the soil. Fungi function to decompose plant tissue such as cellulose and lignin. Mycelia form network around soil particles, giving a soil a crumbly texture. Next is algae. It is only found on the soil surface. Algae can carry photosynthesis. In desert or barren soil, algae contribute in accumulation of organic material of soil. The type of protist found is amoeba and flagellated protozoa. The function of protist is feed on bacteria, help control bacterial population. Viruses in soil mostly infect bacteria. Rare to find fungi and plant are affected. It belongs to Baculoviridae and Iridoviridae group. It is not indigenous to soil but spread by human activity. Virus survival depends on three factors: environmental condition, particular viruses, range from hours to years. Virus is used for biocontrol of insect pests in soil. Where insect pest has been killed, virus tends to disappear. For example, caterpillar stages of celery lupo butterfly can be controlled by the addition of Anagrapha falcifera multiple nuclear polyhedrosis to the soil.
There are some factors that affecting soil microorganisms. The abiotic factors are moisture, oxygen concentration, pH, and temperature. But first, what is abiotic factor? A biotic factor is a non-living part of an ecosystem that shapes its environment. Moisture and oxygen concentration are closely related. Between soil particles, aerobic microorganisms can grow vigorously due to the presence of water and oxygen. Meanwhile, in waterlogged soil, anaerobic bacteria can thrive without the presence of oxygen. Next, pH. Soil pH can vary from 2 to 9. Most soil bacteria have an optimum pH between 6 to 8. Molds grow at almost any soil pH. They thrive at highly acidic soil, so they have to compete with bacteria for the available nutrients. In addition, lime can neutralize acidic soil, thus increase the bacteria population. Fertilizer, which contain aminium salts, provide nitrogen source on plants. When nitrogen metabolized by some bacteria, the bacteria will release nitric acid, which decrease soil pH and increase mold population. Last, temperature. Temperature varies seasonally from below freezing to as high as 60 degrees Celsius as soil surface is exposed to intense summer sunlight. Mesophils are microorganisms that grow best in moderate temperature between 20 to 45 degrees Celsius. They live in warm soil, as an example, Escherichia species. While thermophils are microorganisms that thrive in extremely high temperature between 41 to 122 degrees Celsius. They live in hot soil, as an example, Thermococcus species. Psychrophiles are microorganisms that can survive extremely low temperature, which is below 20 degrees Celsius, and lives in cold area, as an example, Flavobacterium species.
Now, let's get to know about soil pathogens. As we already know, pathogens are disease-causing microorganisms. Here are 4 main human pathogens: Clostridium tetani, Clostridium botulinum, Clostridium perfringens, and Bacillus anthracis. Confused? Don't worry, take your time. Let's get started. Clostridium tetani is a common soil bacterium and can be found in soil, dust, animal intestines, and animal feces, including humans. It can be introduced easily into a puncture wound and can cause tetanus. Clostridium botulinum can produce botulinum toxins under low oxygen conditions. It is often found on fruits, vegetables, and in seafood. It can survive in incompletely processed foods and can cause botulism. Clostridium perfringens, commonly found on raw meat and poultry. It can also be found in the intestines of human and animals. It can cause diarrhea and gastroenteritis. The last one is Bacillus anthracis. It is commonly found in soil of grazing areas and normally affects animals, especially ruminants. It can be transmitted to humans by contact with infected animals or their products. It can cause anthrax.
Alright, now let's turn to the importance of decomposers. At the beginning, let's find out what is the meaning of decomposers. Decomposer is an organism, especially a soil bacterium, fungus, or invertebrate that decompose organic material. Meanwhile, the role of decomposers are they have the ability to decompose organic matter as the flow of energy through an ecosystem, break apart dead organisms into simpler inorganic materials, and making nutrients available to primary products. So, there are four importance of decomposers, which are food chain, nutrient cycling, nitrogen fixation, and ecosystem maintenance. Food chain is a linear network of links in a food web. Organisms in this level of the food chain provide nutrients for the producers, which is plants, who in turn are eaten by the consumers in the next level, such as Bacillus subtilis and Pseudomonas fluorescens. Nutrient cycling is the movement and exchange of organic and inorganic matter back into the production of matter. The plants in the consumer level rely on decomposers to break down dead organic materials to release materials and elements like carbon, oxygen, and phosphorus back into the soil. Moreover, decomposer bacteria are responsible for fixing nitrogen in the soil. They transform nitrogen into a form that can be used by other organisms in the food chain. Some plants like legumes, the bacterium rhizobium lives in nodules on the roots of the plants in a symbiotic relationship. Later, decomposers clean up the dead material by processing it and returning the nutrients to the soil for the producers. It will maintain a balance between living things and resources they need to survive. For the carbon cycle, methane is the main carbon-containing product. It is produced by three genera of strictly anaerobic bacteria, which are Methanococcus, Methanobacterium, and Methanosarcina. In addition to degrading carbon compounds to methane, they also obtain energy by oxidizing hydrogen gas. In one way or another, organic substances are metabolized to carbon dioxide, water, and other small molecules.
Okay, now we go the subtopic under the soil, which is cave. Cave is the place where soil or rock has disappeared. Basically, cave can be formed in four ways. First, slightly acid rainwater can dissolve away limestone. Next, ocean wave pounding against cliff bottoms erode out sea cave, such as in the Fingal's Cave, Staffa, Scotland. Next, streams of hot lava running out of volcanoes cool on their outer surface and harden into tubes, such as Lava Bed National Monument, California. Lastly, microorganisms produce sulfuric acid that eats away rocks. 5% of all limestone caves are formed in this manner, such as Cueva de Villaluz, Mexico. Now, we are focusing on the formation of caves by microorganisms. The wall of the cave are covered with snottites, mucus-like string of bacteria colonies. This snottites or snoticle is a microbial mat of single-cell extremophilic bacteria which hang from the wall and the ceiling of the cave, as we can see in the picture. Microbes eating oil deposit deep below the cave release the H2S, hydrogen sulfide gas, bubble up from the bottom of the cave, forming sulfuric acid when it reacts with water. Plus, these bacteria derive their energy from chemosynthesis. So, what are the relationship between the releasing of sulfuric acid and the formation of the cave? Because of their waste products are highly acidic approaching pH 0 with similar properties to bacteric acid, researchers at the University of Texas has suggested that this sulfuric acid may be more significant cause of cave formation than the usual explanation offered of the carbonic acid formed from carbon dioxide dissolved in water. However, it leaves a telltale residue and a beautiful crystal of gypsum, such as fluorite, world's most colorful mineral, and chandelier ballroom in Lechuguilla Cave. This gypsum chandelier can be up to 20 feet long, are thought to be the largest in the world.
Hello, have you ever heard about Vinogradsky column? No? Don't worry, let's check this out together. These are the only things that you have to know about Vinogradsky column. Vinogradsky column is a miniature ecosystem used for studying microbial communities. These columns are complete, self-contained recycling systems driven only by energy from light. The function of this column is to study a mini microbial ecosystem, as it can illustrate how different microorganisms perform their interdependent roles. The materials needed to create this column are pond mud, water mixed with carbon and sulfur salts, and parafilm. Do you know how does it work? Color changes over several months show us where microbes with different requirements for oxygen and hydrogen sulfide localize. This column consists of two gradients, which are oxygen and hydrogen sulfide. These two gradients promote the growth of different microorganisms. The added organic materials trigger microbial activity that depletes the oxygen in the mud, therefore creates a gradient with low oxygen at the bottom and high oxygen at the top. On the other hand, high concentration of anaerobic bacteria that produce hydrogen sulfide as byproduct of their anaerobic respiration process are at the bottom, therefore creates a gradient with high hydrogen sulfide at the bottom and low hydrogen sulfide at the top. Well done, you did a great job.